Okay, so thank you everyone for joining us today. My name is Allison Barnes and I'm the Vice Chair of Plant Postdocs. This is a group that's been around for almost 2 years, founded by postdocs for postdocs in the plant sciences, just really trying to help each other connect and get to that permanent job position and support each other by helping review applications and sort of like help postdocs advocate for themselves and get more recognition in terms of like giving seminars and things like that. So this seminar series we started specifically for preprints and new publications because we know that postdocs are not as likely to get invited to give formal seminars at universities. So we thought we could create this space to give our own sort of formal seminars for new publications. So today we have two fantastic speakers, Melissa Brito and Leanna Isabito. And so Melissa will go first today. Oh, sorry, before we get started, I just wanted to announce to anyone who's on the call that there is an informal discussion group open for anyone who's participating today. I'll post the link to that Zoom in the chat below as soon as I stop this. And then also that we are now accepting applications for May to August. And that closes on May 7th at 5 p.m. Eastern time. And that link to apply was recently tweeted from our account. So you can find it there, or if you're in our Plant Postdoc Slack group, I'll post that into the Slack as well. So I'm going to stop sharing my screen so that Melissa can share hers. And then I will, so while Melissa is getting set up here, I'll just briefly introduce her. So Melissa's research focuses on how plants perceive and respond to biotic and abiotic stresses. She obtained her PhD from Queen's University in Kingston, Ontario in 2017. Her doctoral work investigated the role of antifreeze proteins in freezing tolerance and resistance against psychrophilic pathogens in cool season grasses. In 2017, she began an NSERC postdoctoral fellowship with Dr. Jacqueline Monahan at Queen's University and focused on the regulation of plant innate immunity. So Melissa's work uses genetic and biochemical approaches to understand how stress response proteins are regulated in order to engineer stress-resilient crops without fitness trade-offs. Melissa recently started a postdoctoral fellowship at Iowa State University, working with Dr. Stephen Whittam to explore the use of viral systems to induce heritable gene edits in monocrop crops. Man, I cannot talk today. Okay, so with that, Melissa, take it away.
All right, thank you, Allison, for the introduction. And thank you again to the Plant Postdoc Committee for providing this excellent opportunity for postdocs to discuss their work and have it reviewed in this platform. So today I'm going to be discussing some of the work that I did during my postdoctoral fellowship at Queen's University. This work was conducted in Dr. Jacqueline Monahan's lab, and it characterizes the subfunctionalization of a multifunctional protein kinase by phosphorylation. This work has broad impacts for the subfunctionalization of protein kinases in plants and also has prospects for engineering disease-resilient crops without having developmental or growth trade-offs. So this work is currently available as a preprint on bioRxiv and will also be available soon on PNAS. So currently, it is estimated that 15% of global crop loss is due to diseases every year, and this amounts to over 100 billion US dollars annually. So there's already a tremendous strain on food and fuel production, and this is only estimated to increase as a result of a growing population and increasing global temperatures. So now more than ever, it's very important to understand how plants respond to disease and to develop new technologies in order to make more resilient crop species. So the ability to defend against disease requires plants to be able to sense and respond to bacterial pathogens prior to their causing disease. So I'm going to be showing a very simplified version of the initial response to immune detection. However, it's important to note that this is very much more complex than what I'm going to be showing and involves a number of different proteins. So sensing microbial pathogens first occurs through the detection of conserved microbial molecules known as microbe-associated molecular patterns, or MAMPs. A classical example of a MAMP is bacterial flagellin, or the 22 amino acid epitope that's recognized by the plant, which is flag 22. So following binding of these MAMPs to these pathogen recognition receptors, we have the activation of the intracellular domain of these receptor complexes, and this is going to initiate immune responses. Now, one of the first responses to pathogen detection is the phosphorylation and activation of intracellular receptor-like cytoplasmic kinases. So one example of a receptor-like cytoplasmic kinase that's required for immune signaling is botrytis-induced kinase 1, or BIK1. And BIK1 serves a number of roles intracellularly that culminate in defense responses. So numerous proteins have been targeted in this innate immune pathway in order to attempt to increase plant immunity. However, the upregulation or overactivation of defense responses often comes with consequences to growth and development. So this is exemplified well if you look at Arabidopsis seedlings that are grown in the continual presence of bacterial MAMPs. You can see that these seedlings are much smaller, and this is due to reduced resource allocation being given to growth and development as a consequence of these continually activated recognition receptors. This can also be seen in plants that have constitutively activated innate immune responses in the absence of pathogen detection. These plants are extremely small, will have necrotic lesions on the leaves, and will often have troubles producing seed. So it's really important for the plant to be able to have an optimal level of immune signaling without having these symptoms associated with the overactivation of these responses. So one way that a plant does this is by limiting the accumulation of that receptor-like cytoplasmic kinase BIK1. So CPK28 is a calcium-dependent protein kinase. As suggested by its name, it is activated in response to increases in cytosolic calcium. Upon its activation, it is able to phosphorylate and activate two E3 ubiquitin ligases, PUB25 and PUB26, which in turn can polyubiquitinate BIK1 and target it for proteasomal degradation. And this will dampen immune signaling. As one might expect from a negative regulator of immunity, CPK28 loss of function plants have enhanced resistance to microbial pathogens. So this is showing bacterial proliferation of the gram-negative bacteria, Pseudomonas syringae. You can see in green here, our two loss of function lines are more resistant to disease than our wild-type Columbia plants. And if we look at vegetative plants, our CPK28 loss of function plants don't have huge reductions in plant growth, and they don't have necrosis associated with these increased immune responses. So this makes CPK28 an excellent target for engineering enhanced resistance. However, it's a multifunctional protein kinase that serves roles in additional pathways, and its downregulation is associated with pleiotropic effects. So CPK28 is also involved in the transition from the vegetative to the reproductive stage. And as a result, CPK28 has secondary growth and developmental defects, such as the stunted stem elongation. So the purpose of this work was to try to determine the pathway-specific regulation of CPK28 in order to make the protein non-functional in immunity while maintaining its role in stem elongation. So calcium-dependent protein kinases are bipartite calcium sensor proteins. So they have a carboxyl terminal calcium binding domain. So you can see here 4 EF hand motifs. These are negatively charged and allow the protein to bind positively charged calcium ions. They also have an amino terminal kinase domain. So these kinases are able to sense changes in intracellular calcium levels, and then they're able to propagate signals through initiating the appropriate signaling cascades. Between the kinase domain and the calcium binding domain is this autoinhibitory junction, or AIJ, and this region is required for conformational changes that are needed for activation. So when calcium levels are limiting, most CDPKs take on an inactive conformation. So in this conformation, you have the autoinhibitory junction in the calcium binding domain, and they're physically occluding the active site of the kinase. And this is due to a lack of calcium loading in the calcium binding domain. And then with an increased availability of calcium in the cytosolic space, you get this dramatic conformational change that pulls the AIJ and the calcium binding domain away from the active site, allowing the kinase to be active. So in this way, calcium-dependent protein kinases are thought to be activated by stimuli-dependent calcium signatures, and then they're able to decode those responses by initiating the appropriate signaling cascades. However, CPK28 is a multifunctional protein kinase, which means that it would have to be able to decode unique calcium signatures in these different pathways. So it's likely that it is regulated beyond calcium binding. So in this work, we explored site-specific phosphorylation as a regulator of pathway-specific activity on CPK28. So we took known in vivo phosphorylation sites on serine and threonine residues, and we ablated those to non-phosphorylatable alanine residues. We then took these individual phosphoablative mutants and we expressed them in the CPK28 loss of function background. We then assessed these lines for the ability of these CPK28 mutants to complement the loss of function phenotypes. So here I'm just showing as an example, if we ablate serine 495 to a non-phosphorylatable alanine residue and phosphorylation is required for stem elongation, we won't see that restoration in stem growth. In contrast, if it's not required for stem elongation, this will function like a wild type CPK28 protein and we'll see that restoration. So using this genetic complementation screen, we identified one phosphorylation site that was able to direct pathway-specific activity of CPK28. So here you can see on the right-hand side, two CPK28 proteins that are ablated at serine 318, and they're able to restore the stunted stem elongation in this loss of function background. In contrast, this phosphoablative mutant is not able to dampen immune signaling. So you can see here in white are Columbia plants, and in gray, when we put in a wild type copy of CPK28, we're able to restore that dampening of immune signaling. In contrast, on the right, we see two lines here that are ablated at our serine 318 residue, and you see that these plants are more resistant to infection. So what we've done here is we've functionally downregulated CPK28's role in immunity in order to provide more disease resistance while maintaining CPK28's function in stem elongation. Now, serine 318 could regulate CPK28 function and immunity by changing subcellular localization or its ability to interact with or phosphorylate biological substrates that are required for its immune function. So in order to test this, we looked at subcellular localization of our phosphoablative mutant. CPK28 needs to localize to the plasma membrane in order to associate with BIC1 and facilitate its turnover. So you can see here the very crisp lines at the plasma membrane surface that CPK28 as well as our phosphoablative mutant are both strictly localized to the plasma membrane. We also conducted in vitro kinase assays using PUB25 as a substrate for CPK28 on the left or serine 318, a mutant on the right. And you can see that both of these kinases can still phosphorylate PUB25 as well as PUB26. So phosphorylation isn't directing immune activity through changes in localization or substrate binding. Now, a recent collaborator had shown that phosphorylated CPK28 requires less calcium for its activation. So we wanted to investigate whether or not serine 318 phosphorylation was required for calcium binding. So in order to do this, we did in vitro autophosphorylation assays with varying amounts of calcium. So either in the absence of calcium, under basal levels of calcium, or saturating levels of calcium. So what you can see here in the left-hand box, this is showing protein autophosphorylation of CPK28 under basal levels of calcium. So even with very low levels of calcium, wild-type CPK28 is incredibly active. However, as you can see in the right-hand box, when we ablate that serine 318 residue, we have a dramatic reduction in activity. So what this says is that phosphorylation at this site is required for calcium activation under these low levels of calcium. In contrast, when we have saturating levels of calcium, so this is 100 micromolar of calcium chloride being added, we see a restoration of this autophosphorylation activity. So what this suggests is that phosphorylation at serine 318 is required for the kinase to be active when calcium levels are limiting. Now, when we conduct these assays over longer time intervals, so over 30 minutes or 1 hour, we see a complete restoration of CPK28 activity. So it's as active as wild-type CPK28. And this is in line with our biological observations. As one might expect, CPK28 would have to be highly responsive to changes in pathogen detection in order to prevent the overactivation of immune signals. In contrast, CPK28 would not have to be as rapidly active during slower, more processive programs such as stem elongation. So in this way, we were able to downregulate CPK28 function by making it less responsive to these low levels of calcium during immune signaling. Now, in order to determine if total protein phosphorylation could compensate for the loss of serine 318 phosphorylation, we did calcium activation assays on phosphorylated proteins or in situ dephosphorylated proteins. So if you look here in this purple box, this is showing calcium activation in gray in the absence of calcium, and then in pink and green with 0.1 or 1 micromolar of calcium. So this is in the physiological range that you would expect inside of a plant cell. Now, if we fully phosphorylate CPK28, we see it's much more responsive to calcium addition. However, if we fully phosphorylate CPK28, but we ablate our serine 318 residue, we see a loss in that phosphorylation calcium priming. So this suggests that specifically serine 318 is required for calcium priming. So when we model CPK28, we find that serine 318 is located in the kinase domain, but it's directly next to the autoinhibitory junction. Specifically, you can see this uncoiled region here, and this is the flexible part of the junction domain that's required for those conformational changes that allow the kinase to be active. So we hypothesized that phosphorylation near this flexible region could be resulting in a conformational change that either results in more efficient calcium binding or is able to stabilize the active state of the kinase. So in order to test this, we did intrinsic tryptophan fluorescence spectroscopy, and this allowed us to monitor changes in calcium-induced conformation. So if you look here in this box, you can see that when you add 100 micromolar of calcium chloride, you get a change or a shift in the spectrum. And this is associated with a change in CPK28 conformation as it binds calcium and frees the calcium binding domain from the active site. In contrast, if CPK28 is fully phosphorylated, we no longer get that shift in spectra when we add calcium chloride. So this suggests that phosphorylated CPK28 is in a conformation that's closer to the active state of the kinase. Now, if we do these same experiments, but we ablate serine 318, we find that regardless of whether or not CPK28 is phosphorylated, we still get that shift in conformation. So what this tells us is that when dephosphorylated, CPK28 undergoes a conformational change when it's bound to calcium. And this is going to allow the kinase to be active. But when phosphorylated, CPK28 takes on a secondary structure that either allows it to more effectively bind calcium or is going to stabilize this active conformation. And this is what would allow CPK28 to be hyper-responsive during an immune response. Now, we wanted to determine whether or not this calcium priming activity was unique to Arabidopsis CPK28. To do this, we use CPK4. CPK4 is the ortholog of CPK28 rice, and it has conserved roles in dampening immune signaling as well as shoot growth. So you can see here two CPK4 loss of function lines that are more resistant to disease, and two homozygous lines here, you can see they have this reduction in shoot growth. So we produced recombinant CPK4 that was ablated at the analogous site to serine 318, so that's serine 315, and we conducted these calcium-dependent autophosphorylation assays. So this is looking at total protein phosphorylation in the presence of basal levels of calcium. You can see here in the right-hand box that when we ablate serine 315, it's less responsive than the wild-type protein. When we add saturating levels of calcium or we allow these assays to be conducted over long time intervals, we start to see the restoration of CPK28 activity or CPK4 activity. This suggests a conserved role in these CPK28 orthologs with priming occurring at this serine 315 residue. So CPK28 belongs to this small group, subgroup of calcium-dependent protein kinases, group 4 CDPKs. When we mine all of the sequences, amino acid sequences from group 4 CDPKs from all land plants on phytosome, we find that a serine residue is strictly conserved at position 318. In contrast, a serine residue is not found in group 1, 2, or 3 calcium-dependent protein kinases. Now, given the conserved roles of CPK28 in immunity as well as shoot growth in various species, it could be possible to generate more disease-resistant plants by having these ablative alleles instead of completely knocking out the function, resulting in these pleiotropic effects. It could also be possible to engineer enhanced calcium responsiveness by mutating the 318 position in these group 1, 2, and 3 calcium-dependent protein kinases. So with that, I would like to once again thank the Plant Postdoc Committee for this opportunity to present. I would like to give a special thanks to Dr. Kyle Bender. He was a postdoctoral fellow who equally contributed to this work when he was at University of Illinois working with Dr. Stephen Huber. Also, thank you to my supervisor at the time, Dr. Jacqueline Monahan, Alex, Danilyn, Alicia, Danielle, and Kaylin, who were undergraduate students that contributed genetic tools to this project, as well as Catherine Dunning, who is a master's student working in Marco Trujillo's lab. And with that, I will take any questions.
Wow, awesome work. That's so great. I know how difficult it can be to sort of tease out that cellular process as to what's actually happening. So that's just really fascinating. While I wait to see if there are any questions that come into the Q&A, I'm going to take advantage of being the moderator to ask a question that I was thinking about at the beginning. So you talked about that mutating this has pleiotropic effects, and the one that you specifically told in this story was about stem elongation. Are there other pleiotropic effects besides stem elongation that happen when you mutate CPK28?
Yeah, so there's some secondary growth defects. So you see increased lignification and you'll see an increase in anthocyanine production. CPK28 has recently been had roles discovered in abiotic stress as well. So drought tolerance and temperature tolerance. So it's not really known how this mutation would affect those pathways, which would be really interesting to test also.
Yeah, that's exactly what I was thinking, because like calcium sensing happens in so many different stresses that's what I was wondering, if it could play roles in other things.
Yeah, so that's definitely an avenue that's going to be investigated in some different plant species, if this mutation will affect other abiotic or biotic responses as well.
Yeah. Okay, so we have a question in the chat. Where is the subfunctionalization where does the subfunctionalization come in for CPK28?
So I guess what we mean by subfunctionalization is that by mutating this one residue, the function of CPK28 is lost in immune signaling, but it's still able to function in stem elongation. So we're kind of teasing apart its ability to function in these different pathways.
Okay, I think we have time for one or two more questions. Do you see preferential CPK28 serine 318 phosphorylation during immune response versus during stem elongation?
Yeah, so serine, we don't actually know if serine 318 is phosphorylated during stem elongation. We know that serine 318, at least in vegetative plants, is as phosphorylated prior to immune response as it is after immune response. And when we make our phosphomimetic mutants, we don't see any phenotypes associated with its overactivation. So we kind of hypothesize that serine 318 is potentially constitutively phosphorylated or phosphorylated most of the time in a plant, but it's a good question, and we haven't actually investigated that phosphorylation in stem elongation specifically.
Cool. I don't see any more questions in the chat, but as a reminder, we're going to have our informal discussion after the seminar. So hopefully if someone thinks of a question that didn't get asked right now, you can ask it later. So thank you so much, Melissa. That was a great talk. And we will move to Leanna.
Great, thank you, Allison.
I'm going to go ahead and share my screen. I love that there's this University of Illinois connection as well. So let's see, okay.
Okay, so while Leanna is sharing her screen, Leanna Isabito is originally from the Washington, D.C. area and is currently working in the wheat physiology group at CIMMYT, which is the International Maize and Wheat Improvement Center based in Mexico. So she received her PhD in crop sciences from the University of Illinois in 2020, focusing on natural variation in photosynthetic traits in rice. Her work included examining the process of photosynthetic induction, which is what she will be talking about today, as well as evaluating the feasibility of including photosynthetic traits into more conventional breeding programs. Leanna is keen to continue working in the area of plant physiology to understand ways in which crops can be made more productive and climate resilient in the hopes of increasing food security. So with that, I will let you take it away.
All right, sounds great. So yeah, so as kind of mentioned in the introduction, a lot of my work is really focused on this characterization of natural variation of different photosynthetic traits. So even though I'm working now with wheat, which has been a whole new adventure, actually all my work previously was in rice. So I'm going to be talking about a paper that came out at the end of last year that looks at precisely that, trying to figure out what kind of variation exists, especially when we think about photosynthesis in the context of non-steady state conditions. So whenever I start a presentation, and whenever I think about this kind of photosynthesis is sometimes kind of hard to imagine, right? I mean, we look at leaves, but we don't actually see the process going on. So I think it's always great to kind of look at the future, to kind of contextualize the work that we're doing and what we also want to accomplish. So what will the world look like by 2050? So we know that we're going to have a growing human population, that we're going to have about 9.7 billion people by the year 2050. We also know that we need about 85% more primary foodstuffs. And this is partially due to the fact that there are changing dietary preferences, now more of an emphasis on animal products as well. And all of this takes plants in order to feed them as well. We also have limited arable land and resources. In the past, when we thought about making agriculture more intensive, the way that we went about doing this was maybe chopping down an ecosystem or forest, but thankfully we know that we can't really do that so much anymore. So we need to be able to make our limited arable land more effective. And of course, we also have to think about climate change. We have much more volatile climate. We also have increased carbon dioxide, which directly influences the way in which plants photosynthesize and the way that we also produce our food. So when we think about future food demand as well, we know that it's increasing, right? So we have more people, food demand is increasing. So when we look at historically what's happened over the past 50, 60 years, we can see that yields have increased quite steadily. Now, if everything continues to go on status quo, by the year 2050, we can expect food yields for crops such as rice or wheat to reach about these levels if we follow this solid colored line. However, this isn't really in line with the amount of food that we'll actually need to be producing by the year 2050, which is more represented by this dotted line. So right off the get-go, immediately, you can see that there's a very big discrepancy by the amount of food that we will be producing most likely by the year 2050, if everything stays the same, and the amount of food that we actually need to be producing if we want to keep up. So this right here shows the really big challenge that we have to face in the next coming decades. So I think the question with this is, well, why photosynthesis? How does photosynthesis really fit into this picture, right? So this is the equation for yield potential, where yield potential is basically the maximum yield that you can expect to have in a given environment under no limitation of water, no limitation of nutrients, and also no pest pressure. And this is a linear relationship between Q, which is the solar radiation per unit of land over growing season. And we can really think about this as a constant. We can't really change the yield. The only way that we could do that is if we picked up our farm and moved to a completely different latitude. But of course, that's not really something that's feasible for most people. We also have interception efficiency, which is the efficiency of plant leaf intercepting sunlight. So how quickly, for example, a canopy develops and fills out to be able to intercept that light. We also have partitioning efficiency, which is also known as harvest index. And then finally, we have conversion efficiency. And conversion efficiency is primarily mediated by photosynthesis. So this is where photosynthesis really comes into that equation. And it's an essential component of yield potential. Now, when we think about these three different efficiencies or these components within the context of how they've been improved over the past 50 years, we saw great gains during the Green Revolution. So much so that interception efficiency and partitioning efficiency were basically maxed out, or as close to we can get them right now, to their theoretical maximum. And this is mostly due to, again, having plants that have canopies that develop more quickly, that close more quickly, excuse me, and also the creation of, for example, semi-dwarf varieties, where you have a larger percentage of the photosynthetes that are being produced by the plant going into the grain as opposed to the stems or the leaves or the roots. So we can see that we saw huge increases in these different components after the Green Revolution. However, if we think about conversion efficiency, which again, we can kind of think of as, at least for these intents and purposes, synonymous with photosynthesis, we don't really see this large improvement. In reality, it hasn't really been improved at all. So we have this huge area here that's honestly an opportunity and hopefully a way to also increase yields. So some of the primary research questions or things that I think about, at least within the scope of photosynthesis, is does significant natural variation exist within crop species for photosynthetic traits? And if so, what does that variation look like? Is it something that can be exploited? Is it something that we can put into a breeding program and really make use of? Are there differences in natural variation between steady and non-steady state conditions? And this is something that I am going to introduce in just a moment. But at least the way that we've traditionally thought about photosynthesis has been in the context of steady state conditions. So what happens when we look at it in non-steady state conditions that really are more representative of what's happening in the field? And then finally, what limits photosynthetic induction or photosynthesis in general? So it's fantastic to know that there might be a lot of variation. It's great to know what it looks like, but how do we actually use it? And figuring out what these limitations actually look like is a great way to start kind of putting this information to work. So a lot of my research is really focused on optimizing plants, optimizing photosynthesis in response to light, or at least within that context. So why would we want to do this in the first place, right? We look at plants, we look outside, and you probably figure plants are pretty good at dealing with light. You know, they've done this for millions and millions of years. But I think the problem is really in the way that we study photosynthesis. So photosynthesis is dynamic, but most of our understanding of photosynthesis is, as I mentioned, in the context of steady state conditions. And steady state conditions are basically where we have the same amount of fluxes entering a leaf or entering a plant as are exiting that leaf or that plant. But when we think about what's actually happening in the environment, we really don't ever have a steady state condition. This is because we have constant changes in light. We have wind that is causing self-shading within the canopy. We have cloud cover that changes sunflex. So really, it's hard to imagine that a leaf, if ever, is actually in steady state conditions. So therefore, there's kind of this impetus to really understand, well, what does photosynthesis look like when we think about it in its actual either natural environment or agricultural environment? So then this brings me to the question of, well, what is photosynthetic induction? This is really what this talk is going to focus about. And at a really basic level, photosynthetic induction is the process by which a plant begins to fix carbon dioxide once exposed to sunlight after a prolonged period of either darkness or shade. And this sounds really simple, right? It's just like, all right, you have low light, you get a highlight, you have this response. But this is really, honestly, quite different than the way that we usually think about photosynthesis, especially it's just like a snapshot in time. This is a whole process. This is a whole process that goes on over the course of many minutes, and sometimes it doesn't even reach a steady state until after an hour. So we have all these different factors that are playing into this process of photosynthetic induction that can actually limit the process as well. So we have to kind of figure out how do we parse this out and how do we make sense of it. So for example, photosynthetic induction can be limited by the regeneration of RUBP. It can also be limited by the photoactivation of carbon metabolite intermediates, or different intermediates involved in the Calvin cycle, the activation of rubisco, which is activated by rubisco activates, I think adeptly named. And then finally, stomatal opening. So how quickly are those stomatas opening in response to a change in light, since stomatas are responsive to both the intensity of light and also the quality of light. So then again, why optimize response to light? So this graph here is kind of what I'll be showing later on when I get into some of the results, but this is kind of what photosynthetic induction looks like. So along the x-axis is time, so starting at 0 over the course of 45 or over the course of 60 minutes, where we have low light and then this transition to highlight and then low light. So in black, we have CO2 assimilation, which is what we can call photosynthesis. And then in red, we have stomatal conductance. So photosynthetic induction is characterized by a lag in photosynthetic efficiency. So the way that this process has usually been thought of is as a step change, or at least the way that's been modeled, right? So we have low light, we turn on the highlight, and photosynthesis very quickly reaches this new level here. But when we actually measure this, we see that this really isn't true at all. We have this very quick change or response in photosynthesis, but it doesn't really look, and of course, this doesn't seem like a very big area, right? I mean, it's still pretty close to what we're seeing in the response in a plant. But when we think that this process may happen dozens, if not hundreds of times over the course of a day, it really adds up. So for example, we know that in wheat, efficient induction costs up to 21% of productivity. So improving photosynthetic induction to allow more CO2 assimilation, so raising the final state that it actually reaches, and also making it faster, so making this slope steeper, making this response quicker, could allow for more effective plants in response to a changing light environment. Of course, this is only half of the equation. I'm really only going to be talking about photosynthetic induction in this talk, but we can also think about on the other side when we have a transition from highlight to low light, where we then have an immediate response of CO2 assimilation, but then a much slower response by stomata, which then represents potential water loss. So really, we have the opportunity to improve not only the amount of CO2 that's being taken up, so the direct productivity of the plant, but also to improve the intrinsic water use efficiency of these plants as well, by really being able to optimize these different responses to light, whether it's from low light to highlight or from highlight to low light. And then very quickly, and just, I guess, to give kind of an idea of how we actually measure photosynthesis, because I know that for me, when I first started with all this, it seemed really abstract. It's like, how do we do this, right? We can't see this with our eyes. But the way that we actually measure this is through using an infrared gas analyzer to measure gas exchange through pores on the surface of leaves, as we all know, stomata. So as stomata takes CO2 up, it also releases water vapor. So we always have this intrinsic balance between the amount of carbon dioxide that's being able to be taken up by the plant, and the amount of water that's also being released. So the way that we can really measure photosynthesis is that we can create a controlled environment where we control different fluxes such as temperature, carbon dioxide, relative humidity, and then measure these different fluxes as they go in, as they come out, and then get an idea of what photosynthesis looks like in these plants. So for example, this is an infrared gas analyzer, and this is the kind of machines that we typically use to collect this kind of data. So with that, I'm going to kind of jump into some of the results for this too. So this project was looking at specifically photosynthetic induction in 5 leaves. And this is kind of, I guess, the continuation of another project that had looked at induction in vegetative leaves in these different accessions. So this work was originally done at the International Rice Research Institute. And as you can see, these are the kind of growing conditions that we had here. We had selected very diverse lines based off of non-synonymous mismatches for rubisco activase. And as I mentioned before, rubisco activase plays a really essential role in the process of photosynthetic induction. So it seems like a good idea to select different accessions based off of that sequence. So when we actually take these plants and we measure what's going on, we had a smaller subset of 6 lines just based off of phenology and the way that it matched up. But when we measure this, we find significant natural variation in rice for 5 leaves for photosynthetic induction. So this is kind of similar to what we'd seen before, but on a much smaller scale, right? Instead of it being from 0 to 60 minutes, it's over the course of about 15 minutes. Actually, 12 minutes. But what we can see immediately is that there's a really sharp increase in CO2 assimilation, or what we call A, in these 6 different lines. And we have IR64, which in this case was kind of treated, I guess, as a check or as a control. IR64 was a rice variety that was released in 1985 and has been considered incredibly high yielding. It's extremely prolific. I think at one that was being cultivated at one time. So it's a really prolific accession. And then also Ausch 278, which is an Ausch variety, which in the work that had been done previously had been shown to be a very low performer for photosynthetic induction. So that's why these two are highlighted. But as we can see here, there's significant variation for these two. When we take a closer look and we look at the average of CO2 assimilation in the first 300 seconds of induction, we see that we can see a lot more clearly these differences between accessions. And the reason that I decided to focus on the first 300 seconds was because initially it wasn't so much of a question of contrasting maybe steady state versus non-steady state, but really understanding that initial response, that initial transition from low light to high light, and see how we can quantify it. So the amount of CO2 that's being assimilated is all one part of it, no? But we also have to think about the speed of induction. And for the speed of induction, we also found significant differences as well. So this is really exciting because this means that there's potentially the possibility to make this kind of response faster. And indeed, when we looked at this, this trait in more accessions in a previous study, we see even more variation as well. But at least in flag leaves, what we can see is that there is significant difference between accessions for IT50, which is the way that we quantify this process. So it's time in seconds to 50% induction. And then also, so to that 300 second mark, and then time in seconds to 90% induction. So again, this is, if you can imagine, to 50% and 90%. So we have a lot of significant difference here, which is, again, hopefully something that can be exploited in the future if we can figure out a way to measure this in a more high throughput manner. So we know that we have variation for the amount of CO2 assimilation that's undergoing, how quickly it's being assimilated. But water use efficiency is also a really important part of the equation as well, right? Especially when we think about how we want to make our crops more climate resilient for the future. And for this, we also saw significant differences. So again, photosynthetic performance is always going to come in an offset for intrinsic water use efficiency, just because of the balance that you have between the amount of CO2 going in and the amount of water coming out. So the plant that is maybe not the best in terms of how much CO2 is being taken up will inevitably be really great intrinsic water use efficiency. And this is also not surprising as well, because Ash varieties tend to be very water use efficient in general. But we have a lot of variation here. Again, when we take a closer look, we can see that there is a lot of variation that can hopefully be exploited in the future as well. So again, this is kind of like putting the sense, feelers out and figuring out what variation exists and hopefully how we can use it in the future. So that's great. So we know that there's this variation that exists, but what can we do with it more practically? How do we actually understand what's limiting it? So induction flag leaves are limited by biochemistry. This is something that I also showed in work for vegetative leaves as well. But in this way, or in this work, what ended up happening is doing a correction to photosynthesis. So basically, let's imagine photosynthesis, and let's take away the stomata limitation. So if stomata aren't limiting the amount of CO2 that's going into that leaf, what would photosynthesis look like? So this is kind of what this correction to photosynthesis does, where we have A, which is in black, and A star, the correction, in white. And for the most part, there's only a 4% difference or less between the A and A star for all these different accessions, with the exception of Ash, which at the end has a difference of about 13% between the raw photosynthetic value and the corrected photosynthesis value, which is indicative of a higher stomatal limitation towards the end of this induction period, at least in this accession. So this is really important to mention as well, because what we've also found is that in different crops, photosynthetic induction is limited by different processes. So we know that in rice, it's heavily limited by biochemistry, both in vegetative leaves and flag leaves. We also know that in soybean, it's also limited by biochemistry. But in other crops, it might be limited by, such as cassava, it might be limited by stomata. So really understanding what the limitation is then can allow us to make a more tailored approach to hopefully improve this in the future. So let's take a closer look at biochemical limitations. So I know this is a lot going on right now, but if you can imagine the induction curve that I'd shown before, which is just done at ambient CO2 concentration, which is about 400 parts per million. Now, if we measure photosynthetic induction not just at 400 parts per million, but also 100, 200, 300, 600, and 800, we get something that looks like this, right? So this is each at a different CO2 concentration. What we can then do is if we choose a point in time, let's say 100 seconds after the beginning of induction, and we choose the value for CO2 assimilation at each one of these points, we can then take this and assemble it to create something that looks like a CO2 to response of intercellular CO2 concentration response curve, or an ACI curve. And this is, I think, really useful, because then what we can do with a curve like this is we can fit it, and we can figure out if photosynthesis in this case is being limited by either the rate of carboxylation or the regeneration of RUBP. And this can tell us about biochemically what's happening in these plants. So what I ended up doing with these plants as well is then measuring, as I mentioned, induction at these different CO2 levels to then construct what I guess we call a dynamic ACI curve. And what's really interesting about this is that this allows us to look at these different limitations to photosynthesis kind of in real time. In theory, since photosynthesis is being logged in this case every 10 seconds, we could have an ACI curve for every single one of those time points. And we could see it over the course of these 12 minutes. What I ended up doing was choosing 5 different points. We can see how these different ACI curves play out, fit them, and see what's limiting them. And what we ended up finding is that in IR64 and IR278, they're predominantly transport. And this is also good to know as well, because it seems to be something that's consistent throughout the course of the actual induction for these, but also allows us to understand what's happening. Another result that I think was pretty interesting and also lines up to what has been seen before is that we didn't see any significant correlation between steady state and non-steady state photosynthetic parameters. And I said this is consistent because this was shown before in soybean, cassava, and also previously in rice. And I just really quickly want to highlight this or mention it because I think this is really important. And I think it kind of comes to the central idea that it forces us to rethink of how we measure photosynthesis. And if steady state measurements are really giving us an accurate picture of what photosynthesis looks like in these plants. So it's just kind of to kind of reiterate that point. And then finally, one of the last points that we had found here is a lack of correlation between vegetative and flag leaf photosynthetic parameters. As I mentioned before, this is kind of like the second part to a larger project that looked at vegetative leaves, now looking at flag leaves. So the idea was, well, if we look at the same parameters, both in non-steady state and steady state, in flag leaves and vegetative leaves, vegetative leaves tell us anything about how leaves, at least in rice, can produce more than 50% of photosynthetates that are actually used for grain filling. And what we find here is that, for the most part, there isn't a strong correlation or a significant correlation between vegetative and flag leaf photosynthetic parameters, which is, I think, really surprising. With the exception of intrinsic water sufficiency and intercellular CO2 concentration, so IW, UE, and CI. And this, I think, is really exciting because this then creates a possibility for early screening development, but potentially, hopefully on a larger scale. So with that, if you want to read more about this is just kind of like the synopsis of everything. You can check the paper out here in case if it's of interest. And if for whatever reason you haven't gotten enough photosynthetic induction, you're like, wow, this is so cool. I want all the photosynthetic induction. There's another paper that's coming out soon, but it's not out yet, that is looking at photosynthetic induction, different canopy levels, but also across different species of rice, looking at wild rice species and seeing what this can tell us about how photosynthetic induction has changed, perhaps on more of an evolutionary timescale. So that'll be coming out eventually, sometime this year, and Food and Energy Security. So with that, I'd like to do my acknowledgements really quickly. I'd like to thank Steve Long and the entire Long Lab back at Illinois. I'd also like to thank the Photosynthetic Research Unit at UIUC. It's such a supportive group. And actually, Steve Huber is part of it too. And I just feel very lucky in general to be able to have worked with them. And I'd like to thank also the International Rice Research Institute, in particular the C4 Rice Center, which lent me all the equipment, all the space, all the seed, everything to do this work. And then also Robert Cohen, Bonner Klondike, who are really helpful on these projects as well. And with that, I'd like to say thanks, and I'd be happy to answer any questions.
Awesome. It looks like we actually already have a question in the chat, so I won't exercise my abilities as a moderator to ask the first question, and instead I will go to this question that says, do you have any idea of wavelength dependence of induction? Specifically, the question asker is thinking about like indoor environments, indoor growth environments.
Yeah, that's a really great question because the quality of light is definitely going to change the way that these plants respond. So for example, for all these measurements, it was done with 90% red light and then 10% blue light. So you're getting a full response from the stomata. I'm not entirely sure what in an indoor growth environment, what the wavelength might look like there, but I would imagine and I would suspect, especially based off of curves that we've recently done without any blue light at all, that you would get a different response depending on the light quality.
So I'm going to follow up on that then. Is that something that you think, like, if we as plant scientists are doing greenhouse experiments and we think that the traits that we're studying or the biochemistry has some tie into photosynthesis, like, are those measurements that we should be taking and reporting in our papers if we're tying photosynthesis measurements into our studies?
Sorry, wait, so incorporating what?
So
like light conditions?
Yeah, so not just light conditions, but like, is there a way to measure like how much red light, how much blue light, those sort of things, and be, you know, so we're, because a lot of times we report like, oh, I grew this on like a 16-hour day, 8-hour night cycle, right? And maybe we report those sort of things, but we're not reporting like, this is how much red light, this is how much blue light sort of things they got, our plants got.
Reported. I think especially with a lot of the light systems that we have now, at least some of the newer ones, you can control how much of each light is going in. So I don't see why not. And I think with photosynthesis measurements, inevitably inside the cuvette, you're always controlling the amount of light, but also the quality of light and the way that it breaks down. So yeah, I think it's something that's worth. I think on the blue light, you're gonna have a very different response than if you had turned it on.
Yeah.
So.
Sorry, I have my dog. Those pesky work from home coworkers. Let's see, any other questions? I did have another one. So you had on your one slide, you showed that correlation between the steady state and non-steady state, and sort of implied that maybe we should be rethinking how sometimes we're taking photosynthetic measurements. And so I was just wondering if you had any like suggestions or ideas about ways we could change that.
Yeah, I think that this is increasingly kind of becoming the hill that I'm like willing to die on. I really do think that we need to be taking more measurements in fluctuating light environments, because I really don't think that, while steady state measurements, a snapshot, a quick measurement is great, it doesn't necessarily tell us about what an entire growing season looks like. It's really hard to infer from that a process. I think what's tough about these kind of measurements is that they're really time intensive, and that's why they're usually done on a really small scale. It's like each one of these curves might take an hour. And then before that, you also have to do a dark adaptation. So maybe for each plant, it might be 2 hours. So that really limits the amount of measurements that you can take. But I think that there's a lot of different groups around the world that are trying to make the study of these processes a lot more high throughput. So using techniques such as chlorophyll fluorescence imaging over the course of time to kind of get a better snapshot of things such as photosynthetic induction, but also NPQ relaxation, which is kind of like on the flip side of all of this as well. Yeah, so I think people are getting creative. Also with like hyperspectral imaging too, perhaps there's something that can be done over the course of a day or growing season.
Yeah, that all sounds really cool. And definitely, because I think about some of these sort of things too, like when I study plant stress response, how there are all of these different variables that we really should be reporting that sometimes we don't think about reporting. These like minor fluctuations that can really impact how our plants are responding. And so, yeah, like you're saying, if we're talking about photosynthesis, they're not always just sitting in the light all the time. And these sort of snapshots are informative, but maybe don't tell the whole story.
Yeah,
okay,
for sure.
we have another question in the chat. Would it make sense to compare photosynthetic induction during drought to check the real contribution of stomatal opening and closing during this important abiotic stress?
I think so. Off the top of my head, if it's going to be during drought, the stomata are just going to be closed anyway, just to try to conserve that water. I'm not sure. I mean, I think with that, we also have like just kind of a cascade of other effects as well. I think with this, what does convince me, at least in terms of what I've measured so far, is when we look at photosynthetic induction and we think of the intercellular CO2 concentration during the induction process, so non-steady state conditions, and compare it to the intercellular CO2 concentration that we see in steady state conditions. For the most part, the CI values, or the intercellular CO2 concentration in the non-steady state, is higher than in steady state. However, you have a lower CO2 assimilation or photosynthesis value in non-steady state than steady state. So it's kind of counterintuitive, right? So you have more CO2 in one, but you're getting lower photosynthesis, and you have less CO2 in the other, and you're getting higher photosynthesis. And this kind of also shows that it is, I think, more due to biochemistry, since you have an abundance, higher abundance of CO2 that's not being fully utilized, if that makes sense. I think that might have been a little bit roundabout. But I think that's a good question too. And I think that these kinds of questions of now just taking photosynthetic induction and kind of taking it out of the vacuum and putting it in either, well, what does it look like in heat stress? What does it look like in drought? Those are things that people are definitely thinking about right now. And I think are projects that'll be coming out in the next few years. So yeah, it totally makes sense to compare.
I'm not seeing any more questions in the chat. So I think that with that, thank you so much for a second great presentation. We had 2 awesome talks today. I'm going to repost the Zoom link in the chat to everyone. And I just want a reminder that everyone is welcome to attend this informal sort of get to know each other, networking, ask any questions that popped up while you sat and thought about our 2 presentations today. So with that, thanks to everyone. Thank you for our attendees, and we'll move over to the other Zoom channel.